All right, so this is going to be the blood lab. And first we're going to have to talk a little bit about what is blood. So I'll use red letters, blood. Obviously it's the stuff going through your veins and arteries that's transporting oxygen and nutrients and such, as well as other things. It's also got the white blood cells, which are more actually about your immune system. So let's, again, very briefly, blood is two parts. It's plasma, which is the liquid part, and then the formed elements, which are mainly cells. About 99% of these are red blood cells. RBC means red blood cell. They're also called erythrocytes. Whoops, let me spell that properly. Hold on. All right, so there are also white blood cells, which we'll talk about in lecture in much more detail, that are involved with the immune system. And there's also platelets, which are involved with clotting, which we'll also talk about in lecture in more detail. But we'll get to know them a little bit. We're going to talk about blood clotting today. Basically, you all know, if you cut yourself, you start bleeding and then you stop bleeding. There's the more dramatic cuts where you bleed, but even, the smallest blood vessels are very delicate and fine. The capillaries are like literally a squamous cell thick. So you're constantly having vessels just kind of getting moving around. Vessels are kind of tearing that, and you got to kind of stop the bleeding. If you don't have blood clotting happening, you just bruise constantly. Like we'll talk about anticoagulants, blood thinners like Coumadin and things like that. If you take that at too high a dose, you just start, getting horrible bruises just trying to put your socks on, right? So platelets are involved with blood clotting. Obviously, if you cut yourself, but also just from any kind of little tears in the delicate little vessels. So we will spend some time looking at these today, particularly the different types. We'll talk about type A blood, type B. It has to do with little markers on these little cells. So we'll look at that in more detail. The plasma, this is mainly water. This is like 92% H2O. And then it's also just a bunch of dissolved solutes and a bunch of proteins. We're going to go into detail in this in lecture. I don't want to spend time on this now. Mainly what you need to know, though, is blood is a combination of these mainly cells in this liquid. This is why it's a connective tissue. It's formally a connective tissue because it's widely scattered cells in an extracellular matrix. In this case, it's a fluid matrix. So it's a bunch of cells floating around in a fluid. And the ratio we care about, the ratio is typically around 55% plasma to about 45% formed elements. That ratio is the hematocrit. And that's something we're actually going to measure today. To measure hematocrit is going to be pretty easy. It's going to be just like doing the fluid balance and RBC lab where you're trying to separate the cells from the fluid? How did we do that? How did we separate the cells from the solution, that when you had like blood cells in a solution and you want to pull the cells, clump them away?
Vortex.
Say it again.
Centrifuge.
Centrifuge, exactly, a centrifuge. So to get the hematocrit is going to be pretty simple. Basically, we're going to have, and I'll talk about this in more detail, but you have these, rather than big test tubes, you have this very thin little, we call it a capillary tube. And we'll have, let's say this is your finger here. This is just supposed to be somebody's finger. And you prick yourself and you've got blood, and you get the blood going up into this capillary tube. And then you put it in a centrifuge. And when you centrifuge it, there's obviously a little cap so the blood doesn't fall out. After you're done centrifuging, the plasma will be at the top and the cells will all be at the bottom. This is like after centrifuging. And then if you want to figure out your hematocrit, the ratio of the cells to the plasma, all you need to do is measure. So you can just get, what is this, total height? This X, this is the height of the cells. And again, it's mostly RBCs, so it's going to mainly look red. And then hematocrit, oops, not E on there, is just going to be whatever this is, X divided by the total height times 100. That'll give you the percentage of RBCs of the total volume of your blood. So that's part of the lab today, is getting hematocrit. Again, if you were actually in lab personally, you would actually stick a needle in your finger and bleed and get the blood into the tube and spin it. But instead, we're obviously not going to be able to do that. Other thing I should, oh, yes?
Sorry, are we also figuring out how to figure out the blood type? Or
Yeah, I will get there. So this is the hematocrit. It kind of made sense to talk about it first since we're doing this here. The capillary tube that you're using here is a special kind. So you have to think about, going back here, these platelets. These are, platelets are for what we call hemostasis and blood clotting. As soon as you cut yourself and the blood is leaking out of the vessels, it triggers this whole chain reaction, which is going to clot the blood and keep it from, hopefully keep it from bleeding. Which means when we go into this thing where you prick yourself with a needle and you start bleeding and you try to get it up into this tube, if we don't do something extra, the blood is all going to turn to jello, because at the blood clotting, you're going to get all this clotting protein, the fibrinogen, forming in there. So how can you keep blood from clotting?
Heparin?
Yeah, so you use heparin. So heparin is a chemical which inhibits blood clotting. So we're going to use what are called heparinized capillary tubes. So the heparin, these prevent clotting. If we did not use heparinized tubes, You would put the blood, even if it's liquid for a little bit and goes up the tube, by the time you tried to spin it down, nothing would happen because it would all just be jello in there. So if we go back again to here, this can be inhibited by things we call, or maybe I'll go on a new page. So hemostasis, blood clotting. Inhibited by, we can call them anticoagulants. Sometimes people call them blood thinners. They don't actually thin your blood, they just make it less likely to clot. So these include heparin. This includes warfarin, which goes by the name Coumadin. Like if somebody is at risk of a clot that could turn into a stroke or a heart attack, they might be on Coumadin to make their blood less likely to clot. We'll talk about, when we get to the heart next class, we'll talk about like atrial fibrillation or something. People who have AFib, they give them these anticoagulants, so it's less likely you're going to have some clot forming when the blood isn't like flowing normally. What's another thing that acts as an anticoagulant?
Aspirin.
Aspirin. In fact, some people take little bits of baby aspirin every day to try to make them less likely to end up with a stroke, like having some aberrant clot form. Another thing that actually is a really powerful anticoagulant is one of the things people take as an herbal supplement, ginkgo biloba. That we had a few years ago, I had some students do an independent research project looking at the anticoagulant properties of ginkgo and found it was even more powerful than aspirin. Which means like if you're going, you get wheeled into surgery and the docs are asking you like, have you taken any drugs? Are you on this or that? You should definitely, if you take like ginkgo, you should let them know, because if they cut you open, you're going to bleed a lot longer if you've been taking ginkgo than if you hadn't. So we're going to be looking at this a little bit in our lab today as well, because we can time how long it takes from the time the blood starts leaving the body to when the clotting happens. To do this, we're going to actually use a different kind of capillary tube. You'll have a capillary tube like we had last time. And again, capillary tube just means a thin little glass tube, and you'll fill it with your blood. But in this case, since we want to time how long it takes for the blood to clot, obviously we don't want heparin. If we had heparin here, it would never clot. So this has to be non-heparinized. Right? If it was heparinized, you would not ever get it to clot. And blood clotting, again, we're going to talk about blood clotting in more detail in lecture. But the basic idea is there is a soluble protein in the plasma called fibrinogen. And this fibrinogen turns into fibrin. Fibrinogen, which is soluble. So the fibrinogen protein in the plasma is just another dissolved molecule. But then it becomes what's called fibrin. And this is not soluble. This is this fibrous, sticky thing that creates, it's what kind of creates the Band-Aid, the patch, when you cut yourself. So fibrin, this is insoluble. So this is what the clotting happens. When the clotting is happening, this is what's going on. You're taking this plasma protein, this fibrinogen, over the next few weeks, you're going to get to learn lots of different plasma proteins. Some of the plasma proteins are involved with blood clotting. Some are involved with your immune system. Some of them are involved with your blood pressure control, all that. So we're going to see lots of different plasma proteins. But this particular plasma protein, fibrinogen, is involved with blood clotting. During the blood clotting process, when you have a breach of the vessel, it starts a chain reaction, which we'll talk about later, basically initiated by the platelets, which are going to create this fibrin. So what we can do is, take your finger, prick your finger with a little needle. You start bleeding. As soon as the blood is out of the body, we basically start a timer because we've started this chain reaction that is going to lead to the formation of the fibrin. And so quickly, we put that blood into a capillary tube. And then we, like after a minute, you break the tube in half. Snap. As long as the fibrinogen is still soluble, then it's just going to be, liquid blood in both sides. It's just going to break and there won't be anything interesting to see. You do this again after 2 minutes, after 3 minutes. Usually we have the data, so I don't have to, you'll see the data. Usually somewhere around 3 to 4 minutes, you break it and something very different happens. All of a sudden, it's like, holy Toledo. There's actually some gooey, stringy stuff that is spanning the space when you break this thing and try to pull it apart. So this is like clotting has occurred. So you just have to how many minutes in. So that's how you're going to assess the time for blood clotting, is basically you fill up one of these little capillary tubes. Again, this time non-heparinized because you don't want to inhibit blood clotting. And you start a timer. And then you just keep breaking the little tube. And at some point, you've got that thread. And when that thread appears, you know that the clotting has happened. Again, in lecture, we'll go more into detail about the details of hemostasis, there's lots more parts that happen to keep you from bleeding out. But one of the important parts is the blood clot, which happens as fibrin is formed from the plasma protein fibrinogen. So any questions about either of these two things? So these are two of the things that the lab is about. One is assessing blood clotting. And we actually have data from just random students. And then we also have data from students. Usually, as I'm talking about this, I have people like, a few of the people in the class eat some aspirin. So then we can actually see how blood clotting is different when somebody has aspirin in their system as opposed to when they don't have aspirin. So we will look at this both with subjects with and without aspirin on board. We will also be doing hematocrit. Hematocrit is something that is a typical part of just kind of a blood workup. And there's typical values for hematocrit that are normal. When I was saying over here, it's usually like 45% formed elements. That would be a hematocrit of 45%. That's a typical hematocrit. Particularly in guys, it can go up to 50% or a little more. In women, it goes down to more like 40%. Women tend to have lower hematocrits than men. But in your lab manual, it talks about what are typical values for hematocrit for men versus women. All right. So now we're going to get to the red blood cells. Again, red blood cells, aka erythrocytes, who you have met. You're like, oh, I know them. Red blood cell, my old pal from the RBC balance and red blood cells. We're going to look at two different aspects of them in this lab. One, we're going to look at blood type. Heck. Which I will talk about in much more detail in a few moments. We're also going to look at just overall hemoglobin concentration in the blood. Hemoglobin, an RBC is basically a little bag full of hemoglobin. Each red blood cell, one RBC, has around 250 million hemoglobin molecules in there. And these bind and carry oxygen, and also plus some CO2. And we'll talk much more detail about hemoglobin in lecture as well. We'll talk about hemoglobin's job carrying oxygen. We'll talk about its job carrying carbon dioxide. We'll talk about how the binding affinity of hemoglobin for oxygen changes based on different environmental stimuli. So we've got lots more to talk about hemoglobin, but for right now, hemoglobin is just a really important thing to keep track of because hemoglobin is the primary way that you carry oxygen around in your body. And, one of the ways you can have anemia, like the inability to deliver oxygen, is because you don't have enough hemoglobin, usually because you don't have enough blood cells, or sometimes it's because something's wrong with the hemoglobin. So to assess hemoglobin concentration in your blood, there is a more formal way. If you actually do this in an actual clinical setting, they actually have this groovy little thing where you bleed onto this little slide thing, and they put it in this hemoglobinometer, and it shines light in there. And by the amount of absorbance of different frequencies, colors, they can tell how much hemoglobin is in your blood. However, the way we do it in our class is this really kind of cheap and easy way. It's kind of like more of a, get close. We're going to use the Tallquist method to measure. We will measure with the Tallquist method. So again, this is not how they would measure it if you were actually in a more formal setting. This is what you do in a biology lab at school. And the basic idea is you take this, it's like filter paper. Heck, it actually is filter paper. So the Tallquist method is just this. You put a drop of blood on it and you let it dry a little bit. And it turns out that the color of the blood is it goes from brighter red to darker red based on the amount of hemoglobin. So there is this actual cool little chart that you use, and I'll show it to you a little later. You basically then compare it to this chart. And it will tell you like the concentration of the hemoglobin in grams of hemoglobin per deciliter of blood. So is it that color or is it that color? You see, you just match the color. And it will tell you kind of ballpark. It's not great, but it's ballpark. Again, I'll show this to you in more detail in a few moments, but I'm kind of giving you the background of this right now. So this is going to be how we're going to determine hemoglobin concentration. Of the blood. All right, so that is 3 out of the 4 things we're doing. The last thing we're doing is blood typing. The blood typing is going to require a little more background so we have to talk about what are blood types. And then we also have to talk about antibodies, which is actually a good thing to talk about because we're going to have to go much more into depth in antibodies when we do the immune system anyway. So I'm going to start with talking about what are blood types. What does it mean to be type A blood or type B blood or type C blood? There's no type C blood, type O blood. And then I'm going to talk about antibodies because antibodies are a critical part of blood type as well. It's in addition to things on the cells, there's things in the plasma. And the antibodies are also going to be at the core of how we actually figure out your blood type. So the basic idea with blood types. If you remember from the first week of class, we talked about one of the roles that membrane proteins play is kind of ID. The blood types are just these little markers, these special markers on the membranes of the blood cell. So if I have, let's put 4 different things here, and let's make them prettier. So here are some blood cells. In the membrane are these little markers. If I am type A blood, then my markers are this A antigen. And I should mention, so right now, so I'm going to draw it as a little A because it makes it easier. So we have these little A markers if it's type A blood. If I am type B blood, I have a different type of marker. The marker is this one, which I'm going to just, for the ease of drawing, I'll make it look like little bees. So the difference between A-type blood cells and B-type blood cells are they have these identifying markers on their membrane. If I am AB blood, I have a combination. I have both of them. I have A markers and B markers. What about if I'm type O blood?
No markers?
No markers. So an O means there's no markers. So this is the cell. Now what we got to do is talk about antibodies in the plasma. So to do that, I need to talk about antibodies, which I'm sure you all have heard, and there's no way you haven't heard of antibodies. But let's talk a little bit about what exactly are antibodies. They are basically plasma proteins. So they're proteins. They're kind of complicated. They have like a heavy chain and light chain, and they have what's called a variable region and a constant region. Back. So this whole thing is an antibody. And it's got these little things that have a special shape that will bind some particular shape. So this variable region will bind some specific, what we call antigen. So the word antigen comes from the word antibody generating, right? So if I in fact, here, let's give it a little crown. Oh, look, it's coronavirus. Some little nasty little thing, and you want to make an antibody for coronavirus, you're going to end up having the variable region have the exact right shape to recognize that part. So this would be an antibody, against, this antigen. So antibodies have a variable region that will specifically bind onto a particular antigen. So, and again, we're going to be talking way more about antibodies before this class is done. But for right now, you actually do need to understand this in order to understand transfusion reactions and things like that. So this is more of a generic antibody here. I made it against this little coronavirus. But in your blood, if you are type A blood, you will naturally have anti-B antibodies in your bloodstream, just naturally occurring. So anti-B antibodies would grab onto, attack the B markers. Obviously, you don't have any B markers, so that's not going to attach to your own blood. If you are type B blood in your plasma, you are going to have anti-A antibodies. And this is going to be critical when we get to the idea of what kind of blood can you donate to who, or who can receive what kind of blood. Because these antibodies in the plasma will recognize specific things, and again, we'll talk about it more in a few moments. If you are type AB blood, what kind of antibodies would you have?
Neither.
And neither, right? Because if you had type anti-B or anti-A, it would attack your own blood because the cell has both those markers on it. You know, as opposed to O-type blood where you're going to have both. And have anti-A and anti-B. All right, so now we have got to the place where we're going to talk about RH factor, positive and negative, in a few moments. But before we do that, let's talk about the fundamentals of how you can decide whether or not it's safe to give a transfusion or receive a transfusion. So when you're thinking about a blood transfusion, and I should mention these antibodies are going to grab on to whatever they recognize, and they end up clumping up the cells break apart, spills out the hemoglobin. It's bad. The hemoglobin clogs up your kidney tubules. You do not want to have blood cells that are going to be recognized by the antibodies in your plasma. So the core thing you need to think about when you are trying to decide about blood transfusions, think about the antibodies that are in the person's body. Think about what what they're going to do when they see a particular blood cell that comes in, a donated blood cell. When you think about donating blood, you don't think about the plasma. Don't worry about the antibodies getting donated. Think about the cells getting donated. So if you took type A blood cells and you put it into this type B person, what's going to happen?
Attack it.
Yeah, the anti-A antibodies are going to attack the cells and break them apart. It's going to be ugly. You're going to have a transfusion reaction. So if you are type A, what kind of blood can you receive?
Type A.
So you can obviously get type A blood because you are type A, and more type A is all good. What else could you receive?
Type AB.
What would happen?
Type O.
These anti-B antibodies in your plasma, if they saw this, they would bind and they would destroy the blood cells. So AB is not going to be able to go into here because the antibodies in this person's plasma would attack it.
You could get type O.
Type O. Right, type O can go because there's no antibodies on type O, so don't got to worry. If I was type B, what kind of blood could I get?
Type B and type O.
Yeah, B and O. You can give, AB again, no, because my anti-A would grab it. A, no, because the anti-A. So how about if I'm type AB, what kind of blood can I get?
A, B, and O.
Yeah, it can get anything, right? Because there's no antibodies. So it's not going to react. So this, we're going to call this our universal recipient. Which happens to be a lucky thing too, that it's a universal recipient, because the incidence of AB blood is really low in the population, like 2 or 3%. So the chances of you finding more AB blood to have for a transfusion is pretty low, but luckily it doesn't matter. You can get any kind of blood and it's going to be cool. What about donating? Who can O donate to? Who can A donate? Well, actually, O, I forgot O. Who can O get blood from?
O.
O can only get blood from O. Since it has anti-A and anti-B antibodies, if you give it any of these that have markers on them, O is going to attack it. So O can only get O. But fortunately, O is the most common blood type out there. So there's a good chance that there's going to be some O blood lying around for you to get as a transfusion. How about receiving? Who can, donating? Who can O donate to?
Anything.
Anything.
Everyone.
Right, so this is going to be my universal donor. Because there are no markers on here, so it's not going to be attacked by anybody. Who can AB donate to?
AB.
Only AB, right? Because AB, if you gave it to any of these other blood types, they all have antibodies that are going to attack the AB blood. Who can A donate to?
A and AB.
Yeah, exactly. Either AB or to itself. And the same with B. B could go to AB or B to itself. A little later, we're going to do a quiz to, I'll put you in little groups, like in another like 15 minutes from now or so. And we're going to make sure you work through some examples. It's important that you actually kind of understand this idea of who can get blood, who can give blood to who, and how that works. And again, the thing you want to think about what kind of antibodies are in the person receiving the blood, and how are they going to respond to the antigen, the markers that are present on the blood that they are receiving, that they are getting donated to them. So any questions about this? So then we should talk about RH marker briefly. The RH. RH is just another marker. So if I have a blood cell, it could be type A blood. If there's no RH marker, I would say this is A minus. If I have type A blood, and in addition to that, I have the little RH marker, this would be A positive. So it's just another marker. You know, if I was O positive, what would I look like?
You would have the RH markers.
Exactly. That would be O positive. There's no A's or B's, but just RHs. In general, you want to think about the RH as well when you're thinking about what you can donate. The basic idea, so I got a little bit of time. So RH is a little bit different in how it works in terms of antibodies compared to the A and the B. the basic idea here is when we were talking about the A and the B, O markers, if you're type A, you automatically have anti-B. If you're type B, you automatically have anti-A. If you're type O, you automatically have anti-A, anti-B. However, if you are, if you're Rh positive, you do not automatically have anti-Rh antibodies. So let's say here's somebody, and let's say they're O negative. I mean, not positive. If you're positive, you can't have them because you're going to attack yourself. So let's say this person is O negative. So they have O, and there is no Rh on there, but they don't automatically have anti-Rh. So let's say this is the first time they're getting a transfusion. Oh my God, they're bleeding. So we put some O positive blood in them, which is going to be O with the little Rh markers on it. The first time their body sees those Rh markers, they're not going to have, so the first time there's no reaction because they don't have the antibodies there to react with the Rh. But once your body has seen this, it's going to now be primed, and in the future, they do have anti-Rh antibodies. Right? It's like coronavirus, right? The first time you get coronavirus, you're getting sick because you don't have the antibodies to protect you from like getting COVID-19. But once you've had COVID-19, then they can do a blood test and see that, oh, now you have antibodies against that because your body has had a response and you're protected more for the future. So the basic idea with Rh, the first time there is not necessarily a reaction because you don't automatically have anti-Rh, but the second time you get Rh positive blood, if you happen to be Rh negative, you are going to have a problem. So in general, you want to just match, right? If somebody is Rh negative, then they can go into either an Rh positive or Rh negative. Okay. If you're Rh negative, since there's no Rh markers to worry about, it doesn't matter who the person you're donating to. If you're Rh positive, you obviously can donate to Rh positive people because they can't have anti-Rh. This is dicey. This can only happen once. And if they've actually been exposed to Rh in the past, they're going to react against it already. So I don't know, does this make sense?
So technically O negative would only be the universal donor, not O positive.
Correct. O negative is the super universal donor. The reason why people have cared about this is because of pregnancies. And what happens is this. Let me clear this stuff. I need to clear. Here's mama. It's a horrible drawing. Here's a little baby growing inside. So because RH and all the blood type is just genetically determined, you could be RH negative. So let's say the mom is RH negative. Well, let's just make it, let's let her be like A positive. And the baby might be A negative. So it turns out there's not a problem the first time that she's pregnant, because oh, God, I'm doing this backwards. I'm sorry. So the mom is A negative. The baby is A positive. So mom does not have anti-RH because she's never been exposed to it. She goes through the pregnancy, it's all good. This baby comes out, is now growing up, and now she gets pregnant with kid number 2. But the problem is giving birth is all sorts of messy. There's blood everywhere. The mom's blood, the baby's blood are all getting all kind of intertwined as she's giving birth, and her body gets exposed to this A positive blood. She actually creates anti-RH antibodies during that process of giving birth. So the second baby now, these things can actually cross the placenta and can start attacking the baby. So what used to happen is somebody would have subsequent births and their body would actually attack the developing baby because of incompatible RH blood types.
Does this happen before birth, or is it only in the process of birth where the antibodies would attack?
No, it's during the development. So the way they stop that now, they give you like a shot. It's called RhoGAM. This prevents the antibodies from forming. So that way you know that it's all going to be cool for subsequent births. Or,
Is this just a one-time shot, or do you have to consistently get it throughout your pregnancy?
I think they give it to you like during the, because the time when you actually get exposed is during the birth, during parturition. So it's not something, so it's something you get like while you're giving birth. So in that process, your body isn't going to make these antibodies as it's exposed to the fetal blood.
I see, thank you.
Okay, so are there any questions? I actually talked longer than I meant to, but at least we've covered everything. Okay. And all right, so what I'm going to do now is we're going to go into the actual lab module. I'm going to now share. All right, doing the lab. Oh, no, wrong lab. What? Reflex, hearing. There it is, blood work. So we won't.
Hello and welcome to the Bloodwork channel. And I'm here today to tell you a little bit about some safety information that you're going to need as you get ready to do the blood lab this.
Right, so when you're working with blood, it's really important to talk about safety. You know, given the amount of time, I'll talk about this a little more in the future. But blood is something that, in fact, let me do this because it is really important. So I'm going to talk about this directly first. Blood-borne pathogens are things you really got to worry about. They kill you. They are like HIV, which is going to give you AIDS, or hepatitis. If you like certain like Hep C, there's a good chance you're going to die of liver cancer before you die of old age. You really need to be conscious about bloodborne pathogens. So whenever you're working with blood that's not your own blood, you have to be kind of paranoid. You know, even if you're trying to help somebody, you come across somebody who's just hurt themselves and they're bleeding and you're trying to stop them from, you know, bleeding to death or whatever. You don't want to end up getting some fatal disease trying to be a good person and helping somebody. So you really need to keep in the back of your mind, how can I help this person and keep myself safe? So in fact, not in the US, but other, like in European countries, it's actually, you'll get a ticket if you don't have latex gloves in your glove compartment. Because if you end up at a car accident or something and you want to go out and help somebody, you don't want to just go out and do it without protecting yourself. You know, you want to put on some gloves because you could have tiny little cuts or whatever. There's lots of ways that bloodborne pathogens can get in your body. It can come in through tiny little cuts that you don't even realize you have, through mucous membranes like your eyes or breathing in. And depending on the level of danger, you'll have more and more layers of what we call universal precautions. Minimally, you're going to put on rubber gloves. Then you go like put on goggles in case things might splash in your eye. Masks if you're worried you might inhale stuff, gowns if it's really messy. So just kind of putting this out there. If we were in lab, we would be really, really, like this is the only lab over my 20 years of teaching where I've actually kicked somebody out of lab for not paying attention to the, you know, to the protocols, because this is where if you're not safe, you can give you yourself or somebody else some fatal disease. Another part of being safe is the idea of sharps. Whenever you are using like, you know, needles to prick somebody, or little glass tubes to do hematocrits, or we'll see we use glass to do to check blood type. All of that, or needles, think about needles. If you're giving someone an injection, any kind of sharp has to be placed in a special sharps container where it gets sterilized. If you just take a sharp, like a little piece of sharp metal or glass that's contaminated with blood and just throw it in the garbage, you know, it's not just like bad form, it's like unethical. It's like some custodian is going to come by and empty out the garbage and push, you know, take this garbage and put it into the big thing, and it's some little needle is going to slice through the bag and poke them, and then they're going to die of hepatitis or something. You know, so you really need to keep track of your sharps. The sharps meaning, again, anything that can cut you, usually glass or metal and things like that. So, you know, I would be remiss if I didn't, you know, spend a little time just saying it's really serious. And now back to our regularly scheduled program.
This week. First item, this is the lab in which.
So that's the sharps container there, just so you can see it. But in the interest of time, I'm kind of running through this. Okay, So the first thing she's going to do is show how to do blood typing. To do blood typing, I should probably talk real briefly about this as well. To do blood typing, it's pretty easy. Basically, we are going to have antibodies. So if you take your blood, you basically kind of take a glass slide, usually use wax pencils to make a little circle so it's going to keep the blood. You put some blood on the slide. This is just like a little glass slide. And we're going to put a drop of anti-A antibody on this, or antibody solution. We put anti-B antibody solution on the other one. And basically what happens if the antibody is going to recognize the blood type, we have what's called agglutination. So let's say I have type B blood and I have anti-B antibodies. We call this agglutination. All of the blood cells start getting all clumped up because the antibodies grab onto one and grab onto the next and grab onto the next. So this agglutination, this is kind of this clumping, which you can see. You can look at the slide and it looks all clumped up. Again, this is something that you're going to see in your little quiz. So back here, let's say I take my blood, I add the anti-A antibody on this side, the anti-B antibody on this side, and then I take a look, and it looks like this. One side, the A side looks like this, and the B side looks all clumpy. What kind of blood type am I looking at here?
B.
I'm looking at type B blood because the anti-B is clumping because it's recognizing blood with B markers. Exactly. What would it look like if I was type O blood?
They both wouldn't be clumpy.
Right? There'd be no clumping in either one because there's no markers for either the A or the anti-B to grab onto. What if you were type A blood? What would it look like?
The A one would be clumpy. And the B one would be regular.
Yeah, exactly. So this is kind of how you're going to recognize the blood type. You know, the antibody will clump if the blood has those markers. So you know, oh, that's that kind of blood. You know, for the RH, it's the same basic deal, but instead of, you know, anti-A, you just put a drop of anti-RH. And if it clumps, it's RH positive. If it doesn't clump, it's RH negative. Right? So let's go back to
Okay, my number one recommendation for everyone is that you get yourself organized. So you want to have all the materials that you need, out in front of you. You want to know, as soon as you stick yourself, where is the blood going? I can't tell you how many students have so much anxiety over sticking themselves, and then they get a nice stick and they're bleeding and then they don't know what to do. So get everything organized. Okay, for the purposes of this demo, what I'm going to show you is the blood typing for A and B and RH, and also filling a capillary tube for the hematocrit. Those are actually some of the, certainly filling a capillary tube can be a little bit challenging. So what I have in front of me right now is the following. I have two slides, one for the A and the B. I can do that on one slide, so make sure you use your little wax pencil so you know which one was which. And then I need a second slide for the RH. And the reason for that is the RH needs to be put on a slide warmer in order to see the result. I need toothpicks, one for each, so I have a few toothpicks out. And I also have my capillary tube. Capillary tubes are teeny weeny little tubes, and they come in this little container. They're very small. Obviously, you can't see this from there, but one end is red, and I'll show you how to fill this. We'll do a close-up in a second. And then I have my antibodies that you're going to be adding when we do the A and the B and the RH. The last thing in front of me is this. This is called CryoSeal, and this is how we're going to seal the capillary tube. We're just going to seal one end. Okay, most important thing is how to use the lancet. There are a variety of different models that are out there, but they all have the same basic idea. There's a safety on one end. It's a little plastic T, and you're going to pull that off. With the lancets that are at the bookstore, you know that when you cut yourself.
Yeah, I'm going to speed on since you're not going to be sticking, but we actually, at school, we have cool little lancets. And yeah, where do you put the lancet once you're done with it?
Sharps container?
The sharps container.
If you don't bleed very much, you want to try things like shaking down the hand, or the best thing you can do is run your hand under some warm water. You get some nice vasodilation, we'll get some nice blood out of you, and hopefully you can do all this with one stick. Okay, so I'm going to demonstrate that next. Okay, so what I'm going to do is use a little alcohol swab. I'm going to go on the side of my finger where the skin is the most thin, and I'm going to take my lancet. Now mine has a little safety right here at the end, so I'm going to twist that off. And with this one, I caulk it by pulling this part back until it clicks. Now it's ready to go. So I'm going to place this over my finger and I'm going to push the button. Ta-da! I'm going to squeeze. Hopefully I'll get some blood out to show you. Oh, there we go. And I'm going to try and fill this capillary tube. So do you see how it fills? You want to get about 3/4 of the way up to the top if you can. So there I am. Then you're going to take the open end.
Right, so that's a horrible view of it. So imagine and there's an art to it. And again, because we're not actually in lab, it doesn't make as much sense to spend the time on the art of filling a capillary tube, but we'll get back here.
And place it into the CryoSeal. It's like clay, and it's going to seal. And now you can just set it on your bench and wait until your instructor has a full load ready to go. For the A, B, and RH, you want to have already set up the drops. So this is antibody A, this is B, this is RH. Get 3 clean toothpicks. You need a little bit of blood, and then mix a little bit of blood with each drop and look for clumps. There's my A. You can see the clumps already, actually. Take a new toothpick. And now I'm going to test for antigen B. And I'm going to mix that in there. And then one for the RH, again on a separate slide because you will have to warm that one. There you go. Now, I don't know if you can see this already, but both of these are clumping. So you don't need to look under a microscope to see this. You just are looking for the blood clumping and not to be smooth. Now I have antibody A clumping. That means that my blood contains antigen A. I also clump with antibody B, so that means that my blood contains antigen B. The RH you can't tell because I haven't heated it, but that's how you're going to do the A, B, and RH. And that's
California. Let's talk about Prop 22. 22 lets app-based drivers stay independent and provides new benefits, including an earnings guarantee and health care. Join.
Sorry about that. Good God.
Okay, Okay, the last thing that I need to show you is our little desktop centrifuge. And when you pop the lid on this thing, it's got a cover. And this is, believe it or not, the trickiest part of it. And we're going to ask.
Again, she's basically going to put the blood in there and turn it on.
Pull your tubes out after they've spun and get a ruler, and you'll be able to measure the hematocrit, because all of the, what we call hard fraction, all the erythrocytes will get pushed to the perimeter against that cryocil plug, and all the plasma is going to float on top. That's it. As always, I hope that was helpful. Thank you so much for visiting the Penguin Prof channel. Please show your support by.
All right, so you know, we don't have to do these other things right now. These are just kind of more detail. Like I said, filling up a capillary tube is kind of an art. You know, it's worth watching this a little later. And she's kind of showing how you kind of tilt it back and forth. And again, the basic idea is you fill it up, you spin it, and then you can measure the hematocrit. This is worth looking at, this picture here. This is a picture of what the two different kinds of tubes that we use in this lab look like. Which one would we use for doing hematocrit? The one on the left or the one on the right?
Left.
The one on the left, exactly. The one that's heparinized. The one on the right is the one that you use for the blood clotting. So what time is it? We're going to come back here in just a few moments. Let me just finish up here. Hemoglobin content is just really kind of simple. Good God. There's more and more.
Hi friends, this is a quick way to evaluate your hemoglobin in.
So this is the Tallquist. So you can just see what Tallquist is when you're trying to measure hemoglobin content. Remember I said it's like you put your drop of blood on the filter paper and compare the color to the chart. This is just this dude showing it.
The blood. You need to have this paper and the color. Here you can match the color of the strip, the blood. You need to prick up your finger and the blood will come out. You need to put up that one drop of the blood on the strip. The strip will absorb it and its color will change in the course of 30 seconds. And you have to wait for 30 seconds. You now put the drop and then you wait for 30 seconds. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. And so on. You need to wait for 30 seconds. And then after that, you place this strip below that particular card that you see the color coding card there. And below that, you point that in the hole. And then you need to compare it. Now place it below the hole and you to compare it. For example, the 6 color is quite light as compared to the blood that we have in the strip. Now it's nearly equivalent to the 14. So the value is 14 gram per DL. And this is how you can quickly see your.
Yeah, so it's 14 grams of hemoglobin per deciliter of blood. So that's how you do this Tallquist. It's just basically matching the color. So this same guy is doing a coagulation time. Again, this guy, I always feel a little bad showing these. It's kind of always using like a rusty needle to poke himself, but this is what I found.
Hi friends. Today we will.
So coagulation, again, this is using the non-heparinized. This is using the tubes that do not have heparin. So it's going to allow the blood to clot. And we know blood has clotted because fibrin, that thread, will show up.
Try to understand the clotting time, the normally which is around 2 to 8 minutes. The duration when the bleeding starts and up to the formation of fibrin threads is known as the clotting time. In order to do so, clean your finger with spirit and prick the finger. Show that the prick should be bold and the blood comes in the form of drop. Now touch the capillary with the side so that the capillary sucks the blood with the capillary action. The care to be taken so that no air is trapped in it. There shouldn't be any air bubble trapped in the capillary. Now hold the capillary in the palm at the body temperature for around 2 minutes. Then after 2 minutes, break the capillary slightly from one end. And you try to see whether there is any thread formation is there or not. If not, then you have to break it again after every 30 seconds. You have to need to break it again till you get the threads. Now the time when you get these threads is known as the clotting time. This is
So you all see that there's gunk in between. That's how you know. If he's not really showing the earlier one where there's nothing there, but it kind of gives you a sense of what that looks like. So, you know, in this module, there is data on, you know, hematocrit, average hematocrit for male, female, average hemoglobin for male, female, clotting time on average for the normal subjects, clotting time for the subjects with aspirin. So this is all data that you are going to use when you're doing your write-up of, you know, kind of describing, answering the questions and stuff. What I want people to do for the last 10 minutes is there is a quiz. If you go into this module here for part 1 and 2, blood typing, it says Bloodwork typing quiz. If you click on Bloodwork typing quiz, it's basically gonna give you, you know, here's these things. It shows you what do this is a control, just what blood would look like. This is what it looks like with anti-A, anti-B, anti-RH. And you have to decide what kind of blood type it is. So what I want people to do is, and then there's some questions about donating. I want people to do this together. So I'm gonna put you all into breakout rooms again, and I want you to actually talk with each other and in your little groups, do this quiz just right now, because that's going to be the way you're really going to sit with this material and figure it out. So this shouldn't take more than 10 minutes. So we'll come back to say goodbye at 12:30. But what I want to do right now, are there any questions about any of this at this moment?
Sorry, where's the quiz? I'm looking
where's-
on lab 13.
Yeah.
It's in the blood typing label, like part 1 and part 2. If you scroll down to the bottom below the videos, it's right there.
Do you see it now?
No. Oh, there it is. Okay. Parts
Yeah, I
1
see
and
it.
2.
All right, so I'm gonna just automatically pop you into some breakout rooms right now. So here you go.